Dear students, this is our 24th lecture on medicinal chemistry and second lecture on antibacterial drugs. In the first lecture on antibacterial drugs, we have discussed about the mechanism of action in case of inhibitors of cell wall. Whereas in this lecture, we will see what is the mode of action in case of inhibitors of protein synthesis, inhibitors of cell membrane synthesis, inhibitors of nucleic acid synthesis, and inhibitors of metabolic pathways. As I've told you, lecture 1, which has already been uploaded, that is 23rd lecture on medicinal chemistry. We have discussed about inhibitors of cell wall synthesis, and remaining these three we will discuss in this lecture. Now next is inhibitors of protein synthesis. Uh, Protein synthesis in bacteria can be inhibited by several antibacterial drugs without significantly interfering with the protein synthesis of the human cells. The reason behind this is that the ribosomes in case of bacteria and ribosomes in case of human cells, they are structurally very different, and this leads to the selective toxicity between the different types of antibacterial drugs, which will inhibit the protein synthesis mainly in case of bacteria and not in case of human cell. So this selectivity is because of the difference in bacterial and human ribosomal proteins or RNAs or associated enzymes. For example, bacteria, they will have ribosomes which are called as 70S ribosomes, having two subunits, 50S subunit which is called as the larger subunit, and 30S subunit which is called as the smaller subunit. So smaller subunit, this is 30S subunit, larger subunit, this is a 50S subunit. This is present in case of bacteria. Whereas in case of human cells, we call the ribosomes as 80S ribosomes, and here the larger subunit is 60S subunit and smaller subunit is 40S subunit. So they are structurally different from that of subunit in case of bacteria, and because of the structure dissimilarity between the human cell ribosomes and bacterial ribosomes, they behave differently towards these antibacterial agents, and protein synthesis in case of bacteria is affected by these types of antibacterial agents. So as far as mechanism of action of these antibacterial agents is concerned, that these agents can either inhibit the activity of smaller unit, or they can inhibit the activity of protein synthesis in case of larger subunit. So depending upon the inhibition of activity on the type of subunit, different classes of antibacterial agents, they have been classified. For example, tetracyclines and aminoglycosides, they are they act on the smaller unit, so they will inhibit the protein synthesis in case of smaller subunit. Whereas macrolides, lincosamides, and chloramphenicol or oxazolidinones, they will act on the larger subunit, thereby inhibiting the protein synthesis in case of larger unit. Now, the each category of these antibacterial agents, they act differently on each subunit. For example, tetracyclines, what they will do, as I've told you, they will bind to the smaller unit, that is 30S unit of the ribosome, And after binding, what they will do, they will block the binding of tRNA. That is, tRNA binding will be blocked, and this will inhibit the protein synthesis, because this tRNA will help in the synthesis of this growing polypeptide chain. And when the activity of this tRNA is inhibited by blocking of the antibacterial agent with this smaller unit, that means the protein synthesis of the bacteria will be inhibited. Similarly, aminoglycosides, the commonly used antibacterial agents, they are streptomycin, gentamicin, neomycin, and kanamycin. So all these, they will act in a similar way. Mechanism of action of all these aminoglycosides will be similar. So they will bind to 30s subunit of the ribosome, and what they will do, they will impair the proofreading, and this will result in the production of faulty protein. The proteins which are actually required by the bacteria, they are not synthesis, but different type of proteins, they are synthesis. And these different proteins which are not able to synthesize the actual proteins required by the bacteria, they are called as the faulty protein. Similarly, macrolides, common example is erythromycin or azithromycin. These two were the commonly used antibiotics or antibacterial agent even during the times of COVID-19. So these macrolides like azithromycin, erythromycin, then lincosamide, lincomycin, and clindamycin, and chloramphenicol, all these, they act in a similar manner. They will inhibit the activity of the larger unit. So they will bind to the 50s unit of the ribosome, and what they will do after binding, they will inhibit the peptide bond formation. So this is uh, the growing polypeptide chain, and polypeptide chain can grow only with the formation of the peptide bond, and these antibacterial agents, they will inhibit the formation of peptide bond. So therefore protein synthesis will be inhibited. So this is how these types of antibacterial agents, they will act. Then is oxazolidinones. Example is linezolid. This again will bind to the 50s unit of the ribosome, and after binding, they will interfere with the formation of initiation complex between 50s unit and 30s unit for translation. So this messenger RNA, this will be affected. So the initiation complex which is required for the growing polypeptide chain to be formed, that will be affected, and this will inhibit the translocation of the growing protein from the ribosome through tRNA to this. So this is how they will act. Now these are the commonly used antibiotics or antibacterial agents, and this is the subunit on which they will act, and this is how they will inhibit the protein synthesis. And the ultimate will be that they can either be bactericidal or bacteriostatic. Bactericidal means they will kill the bacteria. Bacteriostatic means they will inhibit the growth of the bacteria, but they won't kill the bacteria. So this is the difference between bactericidal or bacteriostatic. So aminoglycosides, they will act on 30s, means small subunit of the ribosome will be affected, and they will block the functioning of initiation complex, and this will cause the misreading of messenger RNA, and they will be bactericidal in nature. Then tetracyclines, they will be bacteriostatic in nature. They will block the tRNA binding to the ribosome, and they will again affect the smaller subunit. Then chloramphenicol, they will affect the all these category of antibacterial agent rather. They will affect the larger subunit: chloramphenicol, macrolides, clindamycin, linezolid, streptograms. All they will act on the larger subunit, and they will block either peptidal transferase, translocation, then peptide bond formation, then early step in ribosome formation or premature release of the peptide chain. So by inhibiting all these types of different positions in the ribosome protein synthesis, they can either act as bactericidal or bacteriostatic. So chloramphenicol is either bacteriostatic or bactericidal. Macrolides, they are bacteriostatic. Clindamycin, bacteriostatic. Linezolid, both bactericidal or bacteriostatic. Bactericidal or bacteriostatic. So the details of all these antibacterial agents: aminoglycosides, they are bactericidal in nature, as I told you already, and they are active against gram-negative rods. Some type of aminoglycosides, they are used against other organisms also. Then aminoglycosides, the name aminoglycoside is because of this component amino sugar linked through a glycosidic linkage to the other sugar derivative. So in all these you can see this is amino sugar, then this is the glycosidic linkage present among these. So that is why these are called as aminoglycoside. And most common examples, they are xantamycin, tobramycin, and amikacin. All these, they are commonly used by acting against the smaller subunit of the ribosome. Then all these aminoglycosides, they have some adverse effects also. For example, these, they show toxic effect on kidneys and auditory and vestibular portion of eighth cranial nerve. Then they are poorly absorbed from the gastrointestinal tract, and they cannot be given orally. Whereas in the spinal fluid, they show poor penetration, these antibacterial agents. So this is again a side effect. Then they are not effective against anaerobics because they require oxygen for transportation into the bacterial cell wall. So these are the common side effects of these types of antibacterial agents. Then is tetracyclines. These are the commonly used tetracycline: doxycycline, minocycline, oxytetracycline, omadacycline. You can see one thing common among all these antibacterial agents is that they have four cyclic rings present in the structure. That is why, because of the four cyclic rings, they are called as tetracyclines, and they have structural similarity also. In all the cases you can see, only difference is some substitution here and there, they are different. Otherwise structure of all of these is same. At some positions some modification or substitution is there, because of which they show different properties towards different types of infections. Then again, tetracyclines, they are bacteriostatic in nature, and they are effective against gram-positive as well as gram-negative bacteria, mycoplasma, chlamydia, and rickettsia. So what they do is they inhibit the protein synthesis by blocking the amino acid transfer RNA. tRNA is affected from entering the receptor site of 30S ribosomal unit. So in general, they have low toxicity, but they are also associated with some important side effect as we have seen in case of aminoglycosides. The next is chloramphenicol. Chloramphenicol acts against larger subunit and they block the action of peptidal transferase enzyme. Again, this is the chemical structure of chloramphenicol, and what it do is that it inhibits only bacterial protein synthesis selectively because it will bind to the 50S bacterial ribosomal subunit but not to the 60S human ribosomal subunit. Then is macrolides. Commonly used macrolides, they are azithromycin, clarithromycin, and erythromycin. All these they have similar structure again. Only difference is and here and there substitution is different here also. So some either hydrolyzed products they also show the same type of activity. They are bacteriostatic in nature and they show wide spectrum of activity. That means they are effective against gram-negative as well as gram-positive bacteria. Macrolide generally refers to large size ring structure from 13 to 16 carbon ring structure you can see over here. So these types of ring structures they are called as macrolides, and you can see these azithromycin, erythromycin, they were also used against COVID-19 infections in recent years. Again, they inhibit the bacterial protein synthesis by binding to the 50S ribosomal subunit and they block translocation of the RNA. So they prevent the release of uncharged tRNA after it has transferred its amino acid to the growing polypeptide chain. Then is lincosamides and oxazolidinones. The lincosamides they prevent bacterial replication by interfering with the synthesis of proteins. And here the mechanism is similar to what we have seen in case of macrolides. Uh, they will bind close to the peptidal transferase enzyme uh, that is 23S portion of the 50S subunit of bacterial ribosomes. Two commonly used lincosamides they are clindamycin and lincomycin. They have similar structure. You can see if this hydroxyl group of lincomycin is replaced by chloro group, then this will be converted into clindamycin. Clindamycin binds to again the larger subunit and this blocks the peptide bond formation. And again this is specific for bacteria in comparison to human 60S subunit of the human ribosomes. The most useful clinical activity of bacteriostatic drug is against anaerobes, both gram-positive bacteria as well as gram-negative bacteria such as Clostridium or Bacillus. Then lincomycin is naturally produced and this can be converted into clindamycin. So as you can see, clindamycin is derived via 7S chloro substitution of this hydroxyl group of lincomycin. Then oxazolidinones, they are new broad spectrum class of synthetic protein synthesis inhibitors. Again, they will bind to the largest 50S subunit in case of bacteria, and they are effective against both gram positive and gram negative bacteria. And commonly used oxazolidinones, they are linezolid and tadizolide. Their structure is different. Linezolid, this binds to 30S ribosomal RNA in 50S subunit and inhibits the protein synthesis. But the exact mechanism of its action is not known. It is thought that it will block some early step, that is initiation complex formation will be affected because of linezolid. Whereas this is advanced version of oxazolidinone, tadizolide. This is second generation drug in the same class of as linezolid, but this is 10 times more active and more effective than linezolid. Then next is streptogramins and other drugs which are used for inhibition of protein synthesis. Streptogramins, what they do is they will cause premature release of the growing polypeptide chain from the larger subunit. So, because of the premature release, the kind of proteins which are formed, they are not the actually required by the bacteria. So that is why the structure and mode of action of streptogramins is different from all other drugs. Other drugs, they will inhibit the protein synthesis, whereas they will form the protein, but the protein formed will be different, which is not actually required by the bacteria. So here the combination of streptogramins are given for different types of infections. For example, the commonly given streptogramins, they are quinupristin or dalfopristin. Combination of these two. This is used for treatment of bloodstream infections which are caused by vancomycin resistant Enterococcus faecium, and it is also approved for use in infections caused by Streptococcus species, penicillin resistance, Streptococcus pneumoniae, methicillin resistant Streptococcus aureus, and methicillin resistant Streptococcus epidermis. So these are effective against these types of bacteria. Then another group of antibacterial compounds is ketolide group of antibiotics. They are also clinically very useful. Telithromycin. This is the structure of telithromycin. These structure is similar to macrolides, uh, but their mode of action is sufficiently different chemically, such that organism resistant to macrolides, they may be sensitive to these types of ketolide group drugs, that is telithromycin. It has shown a wide spectrum of activity against a variety of gram positive and gram negative bacteria, uh, including the resistant pneumococci, which are not affected by macrolides. And they are used in treatment of common community acquired pneumonia, bronchitis, and sinusitis. Then is next is inhibition of cell membrane function. So here only few antimicrobial compounds they can act on the cell membrane. Reason for that is that structure and chemical similarity in the bacterial and human cell membrane is there. So because of the structure and chemical similarity, it is difficult to have an antibacterial agent with selective toxicity. So because of the similarity in structure and the chemical nature of the bacterial cell membrane and human cell membrane. So different types of inhibitors of cell membrane function which are used, they are quite few, and among them polymyxins and daptomycin, they are the two commonly used inhibitors of cell membrane function. Polymyxins, they were first discovered in 1947 as products of Bacillus polymyxa, and they are cyclic peptides which are composed of at least 10 amino acids, and 6 of these amino acids, they will be diamino butyric acid, and these uh, amino acid groups, they will be positively charged and they will act like a cationic detergent, and because of the cationic detergent nature, they will disturb the phospholipid structure of the cell membrane in case of bacteria and human cell also. The most commonly used polymyxins, they are polymyxin B and polymyxin E, which is called as polystin. They are the only clinically useful among the polymyxins. Colistin is active against gram-negative rods, especially Pseudomonas aeruginosa, then Acinetobacter baumannii, then carbapenemase producing Enterobacteriaceae. So these are the this is the structure of colistin and polymyxin B. So here, in both the cases, this substitution R may be H or methyl, whereas in polymyxin B and E, this R1 is different. This R1, if this is benzyl, then it will be called as polymyxin B. If this is secondary butyl, this will be called as polymyxin E or colistin. So this is how polymyxin B and E can be differentiated chemically. The next is daptomycin. Daptomycin again is a cyclic lipopeptide. This is produced by Streptomyces species, and this also work like polymyxins, inserting into the cytoplasmic membrane of gram-positive bacteria and thereby disturbing the membrane and killing the bacterial cell. However, in contrast to polymyxin B and colistin, they which target only gram-negative bacteria, this will target only gram-positive bacteria. So this is how polymyxins, they can be differentiated from daptomycin. So daptomycin is active against gram-positive bacteria, whereas these are active against gram-negative bacteria. The next category is inhibitors of nucleic acid synthesis. Two category of antibacterial drugs. They are effective as inhibitors of nucleic acid synthesis. One category is rapamycins and other is fluoroquinolones. Rapamycins, they will inhibit the synthesis of bacterial RNA, whereas fluoroquinolones, they will affect the activity of DNA. So this is the difference between these two classes of compounds. They will affect RNA, rapamycins, whereas fluoroquinolones, they will affect the activity of DNA, and therefore they will inhibit or block transcription, thereby killing the cell, whereas they will inhibit the DNA replication process and thereby killing the cell. And commonly used drug or antibacterial agent under this category is rifampin, whereas under this category is ciprofloxacin, ofloxacin, and moxifloxacin. The Rapamycins, they have narrow spectrum of activity against gram-positive and limited number of gram-negative bacteria, whereas fluoroquinolones, they show broad spectrum of activity against both gram-positive as well as gram-negative bacteria. Now, these are the structure of different types of classes, rapamycins and fluoroquinolones. The commonly used compound, as I told you, rifampin. This is semi-synthetic antibacterial drugs, and this acts by blocking RNA polymerase activity in case of bacteria. So, RNA polymerase enzyme is structurally different in bacteria and the human cell. Therefore, it shows selective toxicity against human cell or the bacterial cell. And as I've told you, this is used for treatment of variety of infections, but primarily this is given in combination with other antibacterial drugs. So, rifampin, this is the structure. Then quinolones, most commonly used drugs, they are ciprofloxacin, levofloxacin, then ofloxacin and moxifloxacin, they are also commonly used. They inhibit the DNA gyrase activity, and thereby they will inhibit the replication of DNA and thereby killing the bacterial cell. They are effective against both gram-positive or gram-negative bacteria, and they are the most commonly prescribed antibiotics for a wide range of infections like UTI infections, respiratory infections, abdominal infections, skin infections. So they are effective against all these types of infections. Uh, although they are effective against so many infections, but they have some side effects also, like phototoxicity, neurotoxicity, cardiotoxicity. Then glucose metabolism dysfunction can also take place. Then there is increased risk for tendon rupture also if we use these category of antibacterial agents in excess. The next is inhibitors of metabolic pathways. So that is these types of antibacterial agents, they will inhibit the synthesis of folic acid. And two types of drugs they can be used, sulfonamides and trimethoprim. Sulfonamides or sulfa drugs, they are the oldest antibacterial agents, and they are structurally similar to PABA, that is para-aminobenzoic acid. You can see over here, if we replace this carboxylic acid by sulfonamide group, then we will get the sulfonamides or sulfa drugs. So this para-aminobenzoic acid is an early intermediate in the synthesis of dihydrofoliate, which ultimately gives us the folic acid, which is used in the folic acid synthesis. So now when instead of PABA, the sulfonamide is available, then this part will be replaced by sulfonamide. So therefore, the folic acid synthesis will be inhibited. So instead of folic acid synthesis, we will get the sulfonamide mediated folic acid synthesis. And when folic acid is not synthesized, then the natural bases which are required for DNA and RNA synthesis, like pyrimidines and purines, which are required for nucleic acid synthesis, they cannot be synthesized. So, by inhibiting the folic acid, the metabolic pathways they are. So, this is how these sulfonamides they will act as antibacterial drugs. Then is trimethoprim. Trimethoprim again is a synthetic antimicrobial compound. This will serve as an antimetabolite, and this will again inhibit the synthesis of the folic acid, as we have seen in case of sulfonamides. So this is a structural analog of dihydrofolic acid. So here, instead of this component, we will have trimethoprim, and because of that, the folic acid will not be synthesized, and because of inhibition in the folic acid synthesis, the metabolic pathway will also be inhibited, thereby the bacterial cell will be killed. And trimethoprim is used in combination with other sulfa drugs for treatment of UTI, ear infection, and bronchitis. Then next is mycolic acid synthesis. Isoniazid is a prodrug which is used with specific toxicity for mycobacteria, which is used in combination of rifampin and streptomycin in treatment of tuberculosis. So this is the structure of isoniazid, and this again interferes with the synthesis of mycolic acid, thereby inhibiting the metabolism. So these are the references which have been used for preparing this lecture. Thank you very much.